Hi, this is the recording to complete the slides on the lecture on the male reproductive system. So in this title slide, what you can see is a bunch of diagrams that we'll go through as we go through this lecture. This lecture not only goes through the male reproductive system, but also provides a comparison between the male and the female, as well as the development of the reproductive system over the lifespan. So to start off with, the general function of the male reproductive system is to produce gametes called sperm that may unite with the female equivalent of the gamete called oocyte in a process called fertilization in order to form the first cell of the offspring. The other function is once the sperm is produced, it needs to be transported and matured through the male reproductive tract so that it can actually be viable to fertilize the oocyte. So in terms of the general structures, we've got the gonads called the testes, one on each side typically, and these produce the sperm and the androgens, the male reproductive hormones, of which testosterone is the main one, as well as the plumbing or the ductwork or the tubing required to actually mature that sperm. So when we look at this, as I said, the essential organs to produce gametes are the testes. We've got one on each side. These produce not only male gametes called sperm, but as well as the male reproductive hormone, testosterone. And we've got accessory organs to actually support the reproductive process. First of all, we've got the plumbing, the genital ducts, which include, and again, you can see there's two, there's one on each side, the epididymis, vas deferens, ejaculatory duct, and both ejaculatory duct combine to form, transport sperm in through the urethra. And then we've got reproductive glands that help to produce secretions that it transports, nourishes, and matures the sperm as the sperm cell travels through the genital ducts. We've also got other accessory organs, the penis, the scrotum, the spermatic cords that help to transport sperm to outside of the body. Let's start with the scrotum. So as you can see here, the scrotum is a skin-covered pouch that suspends from the perineal region that's divided into two sacs, right? By a septum that's deep to the raphe over here, and each of these sacs contain a testes, epididymis, and the lower part of the spermatic cord. There are two different muscles that help to suspend the testes within the scrotum, and so what happens is the dartos and cremaster muscles help to either lower the scrotum pouch when it's warmer weather, as you can see over here, so that it promotes heat loss, or helps to actually bring the scrotal pouch closer to the body in the instance of cold, threat of injury, or sexual arousal. So again, the distance of the scrotum from the main body differs depending on what else is going on. When we look at the male gonads or the testes, what we can see is that, as I said, the function is to produce both the gametes and the male reproductive hormones, and these testes are active throughout the reproductive lifespan of the male. So starting from puberty up until the death of the person. And the testes are found within the scrotum. Two testes, one on each side, and as it says over here, is suspended by attachment to the scrotal tissue and the spermatic cords. When we look at the internal architecture or anatomy of the testes, what we can see is that there is an outer serous membrane called the tunica vaginalis, and that is a that holds the testes together. And then on the inside, there is the tunica albuginea that helps to divide the testes into 300 to 400 different lobules with the help of the septa. Remember, septa is like a dividing wall. So you've got this septa within the testes, and within each of them, you've got the lobules, and each lobule contain interstitial cells, Sertoli or sustentacular cells, and seminiferous tubules that we'll talk about in a bit. Right now. So when we look at the lobules that are found within the testes, we see that there are tubes, and these tubes within those are called seminiferous tubules. So what you can see over here is you've got this hollow space on the inside. Again, it's called a lumen, similar to other tubes in our body. The space on the inside is called the lumen. And the seminiferous tubule is not only responsible for producing sperm, but also for producing testosterone. And so what happens is you can see that there is quite a thick layer over here. And so what happened is the stem cells of the sperm start from the outer layer, and then as the sperm cells mature, they travel deeper inside towards the lumen, so that when they actually reach the lumen, then they are free to actually be released into the luminal space and to develop as into mature sperm. When we look at the cells of the seminiferous tubules, there's two types of cells. The first one is called Sertoli cells, also called sustentosites, as well as sustentacular cells. And so what these are they are support cells found in the epithelium. So over here of the seminiferous tubules, they provide support and protection for the developing sperm cells, the spermatid. They secrete this hormone inhibin, which secretes a gonadotrophin-releasing hormone that is released from the hypothalamus, as well as follicle-stimulating hormone that is released from the anterior pituitary. So they secrete molecules to promote sperm production. They also produce these androgen binding protein that then bind to testosterone in order to make testosterone, which is a steroid hormone, more soluble in plasma. So these are the Sertoli cells. The other type of cells are the Leydig cells or the interstitial cells. So interstitial means in between. So these are in between cells that are endocrine cells between seminiferous tubules. So we have a seminiferous tubule here, one over here, one over there. What happens is that the interstitial cell will be found in between the tubules. And these interstitial cells secrete testosterone. Next organ is the penis. So the penis is the male organ of copulation. So generally it is flaccid during urination and just normally, and then it becomes stiff and rod-like with sexual arousal in order to penetrate the vagina and deposit the semen into the female reproductive tract. In terms of the structure, you've got the penile shaft, which surrounds the urethra. So if you look over here, the urethra is right over here, highlighted there in the green. And then what happens is you've got 3 different chambers that are longitudinal along the length of the penis. And these chambers get filled up with blood during sexual arousal in order for an erection to be sustained and for penetration to occur. So the 3 chambers run longitudinal along the penis, and then at the end of the penis we've got the gland penis, which is very, very sensitive. So what happens is during sexual arousal, you've got nitric oxide being released near the blood vessels of the erectile chambers. And what nitric oxide does is it causes vasodilation. So instead of the chambers being this small, what happens is it causes these chambers to become bigger, and then blood fills the chambers, and then that causes an erection, right? And so not only does blood actually fill the chambers, what happens is the venous drainage of the penis, blood vessels in the penis, is prevented, and so blood basically stays in the penis. When we move on to the structure of a sperm, the sperm is smaller than most cells. So if you look at the volume of the sperm cells, it's very, very small compared to a mature oocyte. And what we see here is the parts of a mature sperm. So when we look at the parts of the mature sperm, you can see it can be divided into 3 different parts. The first part is the head, right? The head is the part that penetrates into the oocyte. It's very compact. It's got the DNA here, right? It's got the nucleus here that contains the DNA, the haploid DNA, very little cytoplasm. And what happens is at the outer edge of the head of the sperm cell, you've got these acrosome. And the acrosome contains enzymes, which are lysosomal enzymes. And these lysosomal enzymes are released during capacitation in order to facilitate fertilization. So the lysosomal enzymes in this acrosome help to break down the barriers that surrounds the oocyte. The middle part is called the midpiece. And what the midpiece does is it has mitochondria. And if you remember mitochondria, the purpose of mitochondria is to produce ATP, which is an energy source. And so the mitochondria produces ATP in order to actually work the tail part so that the sperm can actually swim. And then the third part is the tail. You can see there's this long end piece, the flagellum, and that helps to propel the entire sperm to actually move up into the female reproductive tract, in through the vagina, the cervix, the uterus, and then through one of the uterine tube, just in case there's an egg there waiting for fertilization to occur. In terms of the germ cells or the sperm cells, what happens is if you look at the order of maturity, so this is the least mature, and this is the most mature. As I said, the least mature cells are found fairly superficial in the seminiferous tubule, and as they become more mature, they get deeper in, closer to the lumen, right? Until when it becomes sperm, they can actually be released into the lumen in order to mature some more as it travels through the tubing that is most of the male reproductive tract, male reproductive system. Spermatogenesis is the production of sperm. So genesis, production of new. Spermato refers to sperm. And as I said, it starts at birth and continues until death, slows down after the age of 35 years old, and also is inhibited with smoking. The process from forming a sperm cell all the way to a mature sperm takes about 64 days. And in terms of the production rate, there's 100 to 300 million sperm produced daily, right? This is very, very at odds with the production of mature oocyte that occurs once every 28 days or every month or so. In terms of the number of chromosomes in the germ cells, what happens is spermatogonium, sperm cells, stem cells contain diploid number of chromosomes. So they have 46 chromosomes or 23 pairs of chromosomes. And then as they divide, they become haploid. So when sperm is released, mature sperm or maturing spermatozoa is released into the lumen of the seminiferous tubules, they're only haploid, right? They only have one of each of the 23 pairs of chromosomes because they will unite with the oocyte, which is also a haploid, to form the first cell of the developing offspring. So you have 23 from the sperm plus 23 from the oocyte to make a diploid offspring. Spermiogenesis transforms early spermatids to true sperm by reducing the amount of cytoplasm that's found in the head of the sperm and forming the various parts of a true sperm, as we can see in the previous slide, to form spermatozoa or form sperm, right? So what happens is, as you can see, when it's just a spermatogonia, it just looks like any cell in the body. It's round. And then as it goes through the mitosis, meiosis, spermiogenesis process, then it looks like it becomes more and more like the mature sperm. In terms of the pathway of the sperm, what happens is once it's released into the lumen of the seminiferous tubules, it then goes into the straight tubules or the tubular recti, which is not indicated in either the diagram. And then from that, it goes into these rete testes, which is the fine meshwork of tubules, into the efferent ductules, and then into the epididymis, right? From the head to the tail, and then from the epididymis, then it goes into the vas deferens and then out. If you look at the epididymis, you can see that it's coiled. And so if you straighten out the epididymis, it's 6 meters long, not 6 feet, 6 meters long. And at this stage, when the sperm is released from the seminiferous tubule, it only contains about 5% of the final semen volume. So semen contains sperm as well as other fluid. And so if you look at the other fluid that forms semen, what you can see is that most of the fluid is secreted by the ductus deferens, right? So you've got the, so basically what happens is without ejaculation, what happens is the sperm basically stays in the epididymis, and then as when erection occurs and prior to ejaculation, what happens is the sperm is propelled from the epididymis to the ductus deferens or the vas deferens over here, right? So this one over here, and then from the vas deferens, you've got the ampulla that secretes fluid to make up 60% of the semen. And if you look at here, some of the fluid that it secretes is fructose, which serves as an energy source for the sperm mitochondria, right? And then from the vas deferens over here, it then, that's the ampulla over here, it then moves into the ejaculatory duct and then into the prostate urethra, and the prostate gland then secretes more secretion, right, to help with the transport of the sperm out of the male reproductive tract into the female reproductive tract. And so if you look at the prostate gland, the secretions that it secretes is alkaline milky fluid. Alkaline is important in order to actually form a buffer against the acidic vaginal secretions. So, and then it also then secretes milky fluid in order to coagulate the semen within the female reproductive tract so that the sperm within the female reproductive tract can actually pause before making its way through to the uterine tubes. And then the last thing is, as the semen emerges, then we've got the bulbourethral glands, which help to secrete thick, salty fluid to lubricate the urethra and the vagina, as well as to clean urine from the penile urethra. If by lucky chance the sperm then makes its way up through the uterus, in through either one of the uterine tube and into the uterine tube that contains the oocyte, then I jumped the gun here. So with ejaculation, what happens is the sperm leaves the male reproductive system and again enters into the vagina of the female. And what happens is the vagina then has vaginal secretions, which is acidic in nature. And so then what happens is the alkaline mucus in the semen helps to neutralize that acidity. And what happens is you've got hundreds of millions of sperm actually leaving the male reproductive system. But by the time it gets into the uterine tubes, it has decreased in number from hundreds of millions to only a few thousand. And this journey of the sperm up into the female reproductive system, into the uterine tube, can take anywhere between 30 minutes or 2 hours. When the sperm actually then reaches the oocyte, what you can see here is you can see hundreds of sperm right here in the green actually being attracted to the corona radiata of the oocyte. And what happens is that the lysosomal enzymes in the acrosome in the head of the sperm then is released to try to break down not only the cervical mucus, but to also penetrate this barrier of the corona radiata. And then what happens is you can see over here that some of these sperm are successful in breaking through and then having contact with the next layer down, the zona pellucida. And what happens is with that, again, the acrosomal reaction is triggered. And then over here, what happens is the acrosome releases the lysosomal enzyme, and you've got this penetration, right? In terms of fertilization, it's typical that only one sperm is successful, right? So success for one sperm. And then what happens is once the successful sperm enters into the cytoplasm of the oocyte, then the nucleus of the sperm is released into the oocyte, and then a diploid zygote is formed. So what happens is once you have a successful sperm, there are mechanisms to prevent polyspermy, to prevent more than one sperm, that's what poly means, from entering and releasing its contents, its nuclear contents, into the oocyte. Because you only want a diploid zygote. You don't want 3 or 4 sets of haploid chromosomes being released, right? So what happens is once one sperm is successful, then you've got the fast block and the slow block. So the fast block happens, as I said, when the first sperm manages to enter into the oocyte cytoplasm, and then the membrane of the first sperm fuses with the membrane of the oocyte. Then what happens is you have an instant change in the sodium ion permeability. Oocyte plasma membrane depolarizes, and it stops other sperm from actually entering through. So it's a fast block because it happens pretty much instantaneously. So that only lasts for a minute, but that minute is enough time for the second or slow block cortical reaction to get triggered. So the slower block takes a few seconds to come about. And so what happens is calcium ions enter in, the cortical granules fuse with the cell membrane, and then you've got the ZIP, the zonal inhibiting proteins, being released into the space between the plasma membrane and the zona pellucida over here. And then that prevents other sperm from binding to the zona pellucida. And so the sperm that are hanging on are released, and then the oocytes, sperm receptors are destroyed. The other thing is that there are mucopolysaccharides that are released to actually harden this barrier, right? To form an impenetrable barrier so that you don't have any more sperm entering in. All right. So we've gone through fertilization there. So we're going to take a step back and look at the functions of testosterone. So one of the big functions of testosterone is to produce sperm. The other thing is to promote the development and maintenance of the secondary sex characteristics, the development and maintenance of the accessory organs, and adult male behavior, including libido. It also helps to regulate metabolism in terms of stimulating growth of protein so that skeletal muscles and bone grow appropriately. And it also promotes the closure of the epiphysis, right? The epiphysis, or the epiphyseal line over here, is the area of growth of long bones. And so what happens is if a person matures early, then it leads to early epiphyseal closure so that the person stops growing in height. But if it's delayed, then that allows the person to keep on growing in height. It also helps to regulate fluid and electrolyte balance, similar to aldosterone that's released by adrenal cortex, but it has a milder or a less potent effect. In terms of the control of testosterone, it involves negative feedback. So the negative feedback involves if there's too much testosterone being released over here, what happens is it sends a signal to the hypothalamus to reduce the amount of gonadotrophin releasing hormone released by the hypothalamus, and then that then also reduces the amount of FSH and LH released by the anterior pituitary, and then that slows down the release of testosterone from the testes. When testosterone release is increased too much, it also then releases the hormone inhibin, and then what happens is inhibin then also inhibits the release of FSH, follicle stimulating hormone, right? Remember, FSH also is involved in releasing androgen binding protein, and it's the androgen binding proteins that bind to testosterone once it's in the hormone to allow a higher level of testosterone to be transported in the blood. When we look at males versus females in terms of the production of gametes or gametogenesis, what we see is here. The start of gamete production in both males and females start during puberty. But the end of gamete production in females ends with menopause, whereas males can produce sperm until they die. And so if you look at the ages of when menopause occurs around 50s, and when puberty occurs around in the teens, what happens is a female can produce female gametes around 35 years, whereas a male, assuming a life span of around 75 or 80 years, can actually produce sperm for around 65 years. In terms of the quantity that male and female gametes are produced, females produced a mature oocyte once every 28 days or once every cycle, whereas males produced anywhere between 100 to 300 million sperm a day. So again, the quantities are tipped in the favor of the sperm in terms of quantity. Timing of mitosis in females, it occurs before birth. And in males, it occurs after puberty. Timing of meiosis 1, it occurs after puberty. In males, it occurs as part of the continuum after puberty as well. And the timing of meiosis 2 in males, again, it's part of the continuum when sperm cells are produced after puberty in order to produce the 100 to 300 million. Whereas in female, the timing of meiosis 2 only occurs if fertilization is successful. In terms of the amount of time that an oocyte is viable once it's released into the uterine tubes, it's only viable for a very short period of time, maybe 24 hours, maybe a little bit longer. And the sperm is viable for about 3 to 5 days when it enters into the female reproductive system. And so you've got the system where it increases the chances of the male gamete to actually be successful in fertilizing the oocyte. In terms of sexual differentiation, I'm not sure whether you know, but we're born with bipotential gonads. So we're born with developing undeveloped gonads that could develop into either male or female. And in the embryo, right, these are connected to a central cavity via Mullerian and Wolffian ducts. As I said, females are considered the fundamental sex. So that means that unless there is a burst of testosterone somewhere during the developmental time, weeks 9 to 12 of the fetal period, the fetus will develop as a female, right? So development as a male requires a functional sex-determining Y region on the Y chromosome that then actively recruits genes to develop into the testes, as well as producing testosterone to differentiate the bipotential gonads into the male reproductive tract. The SRY gene also suppresses genes that are important in female development and differentiate the germ cells in these bipotential gonads into spermatogonia. Otherwise, without the functional SRY gene, then the bipotential gonads develop into ovaries, which then develop oogonia or oocyte stem cells. So what happens is the bipotential gonads in females develop into ovaries, whereas in male, it develops into the testes, right? So you've got that. And then what happens is in females, the Mullerian ducts develop into the uterine tubes and the uterus, whereas in the developing male fetus, the Mullerian ducts degrade. However, in the developing male, the Wolffian ducts then develop into the epididymis, and the vas deferens, whereas in the developing female, those same Wolffian ducts degrade, right? And in the female, what happens is that the cloaca, the cavity, then develops into the urinary bladder, urethra, and the rectum, whereas in the male, those develop into the urethra and the rectum. So again, you've got the split depending on whether the developing fetus has a functional SRY gene or not. So that happens in utero, and then at puberty, you've got further sexual development. So between birth and puberty, in early childhood, there is no sexual development because there's no release of gonadotrophin-releasing hormone from the hypothalamus or luteinizing hormone or follicle-stimulating hormone from the anterior pituitary, right? And then what happens at around 8 to 9 years old, before puberty, the hypothalamus starts releasing GnRH, and that release of gonadotropin-releasing hormone from the hypothalamus then stimulates release of luteinizing hormone and follicle-stimulating hormone from the anterior pituitary. And over here, you can see that in males, it helps to trigger testosterone production. And then in females, it helps to trigger estrogen production, right? So before puberty, the negative feedback loop between GnRH, LH, and FSH is very sensitive. And then what happens is during puberty, that negative feedback loop becomes less sensitive. And so that allows the LH and FSH production to increase. And the gonads become more sensitive to LH and FSH, so the gonads can enlarge and mature and start with the production of sperm in the males and the continuation of oocyte development in females. And then with the decrease in sensitivity to the negative feedback loop, then the sex hormone levels start to rise. So estrogen, progesterone start being secreted in noticeable amounts. Testosterone also does in the male. And then you've got the development of secondary characteristics triggered by testosterone in the male, and female secondary sex characteristics triggered by estrogen and progesterone in the female, right? So this is this whole cycle here that occurs during puberty. Signs of puberty are as follows. So you've got the development of the secondary sexual characteristics, as well as a definition for what puberty is. So it's a stage of development at which individuals become sexually mature, that they can actually continue on with the production of gametes in female and start the production of sperm in males. And then you've got the growth spurt that's associated with puberty as well. And the first sign of puberty in males is the growth of the testes and breast development in females. And you can see over here, development of sexual characteristics include growth of body hair, right? Axillary and pubic hair in females, and the same and just general body hair and facial hair in males. When we look at the amount of hormone that's produced throughout the lifespan in females, what you can see is over here, you've got the amount of estrogen excreted in urine, over here, as well as the amount of estrogen in women. And what you can see here is between birth until puberty, it's pretty much zero, right? So if you can see over here, no estrogen being secreted in early childhood. And then what happens during puberty is the levels of estrogen being secreted goes up. And then it goes up and down during each reproductive cycle. So you can see here, that's why there's a thick band. And then as menopause approaches, the levels of estrogen drops until during menopause, it goes way down. And then after menopause, it drops to virtually, again, nothing, right? So that's what happens over the lifespan of a female. When you look at the levels of gonadotrophins, so gonadotrophins are LH and FSH that's produced by the anterior pituitary. What you can see is that these levels are relatively low throughout, even though it does fluctuate with each cycle. And then during menopause, when estrogen levels drop, then FSH and LH levels increase. And that's because it increases in response to that decline in estrogen, right? So when estrogen levels drop, what happens is the anterior pituitary then starts secreting more LH and FSH in order to try to increase estrogen release. GnRH is also increased, except we can't actually monitor that, but we can monitor the level of FSH and LH. So if you look at the last diagram on the right over here, you can see here early childhood, no LH and FSH release. During puberty, it goes up. During the reproductive years, it then goes up. And then after menopause or at menopause, it goes way up in order to try to increase this decline in estrogen release, right? In terms of male, you can see it's fairly steady because males produce testosterone from puberty until the day that they die. And so what you can see here is this is the hormone production throughout the lifespan in males. And what you can see here is that the testosterone production rises briefly in the fetal period to stimulate the early development of the male sexual organs, as we talked about before. And then at birth, again, there's a brief spurt of testosterone to facilitate the testes to descend, to go down into the scrotum. And then during early childhood in C, there's virtually no testosterone release. During puberty, it starts going up to support sperm production. It continues on. So the red line is testosterone. And then what you can see is sperm production is the blue line. So sperm production starts during puberty and keeps on continuing until the day that the person dies. And you can see that it mirrors the secretion of testosterone. So somewhere towards the person's 50s or 60s, testosterone levels will decline. And then shortly after, sperm production will also decline. So you can see that it doesn't decline to nothing, it just gradually decreases. And this is also indicated in the middle diagram where you've got C, early childhood, D, puberty, and then E, in advancing age, you've got the decline of testosterone levels. And then because testosterone levels just decline and don't go down to zero, what happens is LH and FSH level increases, but not too much, right? Because testosterone is still being secreted. Anyway, we'll talk more about that in class, I'm sure.